video, we're going to look at how to determine the specific activity of salivary amylase. You see specific activity, it is defined as the number of enzyme unit per ml to the total protein, which can be expressed in milligram per ml. Let us take a look at the required materials: phosphate buffer, substrate, activator, inhibitor, coloring reagent, enzyme, coop reagent, distilled water, Folin phenol reagent, standard molecule solution, standard protein solution, pipette, test tube stand, boiling water booth, and spectrophotometer or colorimeter. So first we look at the standard calibration of maltose. Activator is sodium chloride, inhibitor, sodium hydroxide, coloring agent, DNS, 100 milligram of maltose dissolved in water, distilled water, for standard calibration curve measurements. So these can look at the tabular column, the weight to the standard solution from 0.1 ml to 0.5 ml. The activator, inhibitor, and the reagent all added, incubated, and measured at 520 nanometer. In the meantime, we can look at what is these classes of enzymes, or usually these salivary amylases or enzyme comes from the category of hydrolases. Usually hydrolases catalyze reactions in which the cleavage of bonds is accomplished by adding water. Example, when you say salivary amylase, it's hydrolyzes starch into sugars. Similar class of enzymes are phosphodiesterases, which cleaves phosphodiester bond, phosphatases, which remove phosphate group from a substrate, peptidases, which cleaves amide bonds, such as those in proteins. So what actually are enzymes like salivary amylase? So enzymes, are biological catalysts that increases rate of chemical reaction without itself being changed in the overall process. You can see almost all cellular reactions or processes are mediated by enzymes. Enzymes have properties that make them unique. Most enzymes are proteins, not all. Enzymes are highly specific, such as even in this case of salivary amylase. Enzyme exhibit enormous catalytic power. How does it increase the rate of reaction? It decreases the activation energy. It changes the rate of reaction, but it never alters the equilibrium. There's no effect on the position of equilibrium. So enzymes can be simple enzyme or conjugated enzymes. You see conjugated enzyme, they contains a non-protein group called cofactor, which is required for biological activity. So that you can see some cofactors are simple metal ions, other cofactors are complex organic groups. Example, coenzymes, or which is called coenzymes. Some examples can be the vitamin B complex and the coenzyme form. Now again, coming to the video, you can see this, the DNS is added. Which is a colored reaction, red at 620 nanometer. So usually in these assays an activator is used, an inhibitor is used, and a coloring agent used, which can be red using a spectrophotometer. The visible range, thus in a colorimetric or spectrophotometric method assay, you can see the developed color is read colorimetrically at 520 nanometers. At 520 nanometer, in case so based upon the color developed, the concentration of maltose, the higher the color is, the pink color, the higher is the concentration of maltose. So standard as the concentration of maltose used increases, the value in the spectrophotometer at 520 nanometer increases. So you can see that now after calibration, the calculation, so 0.5 ml correspond to 0.0004, 0.4 ml of OD correspond to 100 microgram of maltose, so 20 ml of enzyme liberates the following. The graph, concentration of maltose, okay, it's concentrate OD at 520 nanometer. The straight line that now the estimation of protein. So basically fall in phenol reagent, the alkaline copper reagent, which is Lowry's reagent, the famous Lowry's reagent, the Lowry's method, copper sulfate, so sodium potassium tartarate is used. So actually this Lowry's method, it is read out at 620 nanometer. The DNS method of maltose is 520 nanometer. So for protein, Lowry's method, 620 nanometer. The color developed, the blue color developed, is measured spectrophotometrically. This method is age-old method, used widely, even today, even in this day and age. This is the most followed method of protein estimation. So it was a pioneering work by Lowry a long ago. So you can see the tabular column clearly shows how to estimate. BSI bovine cerebellum is used as standard. The protein, the enzyme, alkyl copper reagent, incubation at for 10 minutes, followed by the addition of phenol reagent, which again incubated for 10 minutes after being read out at 620 nanometer. So green color, blue color for protein estimation using Lawrence method at 620 nanometer. So the DLS method, it is 520 nanometer. So because when you look at enzymes employ several catalytic strategies. Even the salivary amylase enzyme catalytic strategies, different strategies like acid-base catalysis, which involves proton transfer, covalent catalysis, which involves the formation of a reversible covalent bond between, for example, salivary amylase and its corresponding substrate. So what actually the enzyme, how does the enzyme operates? The substrate binds to the active site of an enzyme by multiple weak non-covalent interaction like hydrophobic hydrogen bond, ionic interaction, or reversible covalent bonds. The free energy thus released in the formation of large number of weak interaction between enzyme and substrate, termed form a complex, and because of the energy released known as binding energy. So this enzyme substrate complex is formed so the recognition of enzymes substrate by enzymes is accompanied by conformational changes at active site, and such changes result in the formation of transition state. So when we look at the video here, we are estimating the formation of blue color for estimation of proteins as per Lowry's method. After estimation of protein at 620 nanometer, we are going to find out the specific activity of salivary amylase. So the enzyme substrate complex formed, like here, describe the binding process. There are different models proposed. One model is the lock and key model, which proposes a high degree of similarity between the shape of the substrate and the geometry of the binding site of the enzyme. And another model called induced fit model takes into account that proteins have some three-dimensional flexibility. That is, the binding of substrate causes conformational change in enzyme that results in a complementary fit once the substrate is bound. So we are just estimating the protein. Now you can see the calculation. 0.5 ml of diluted enzyme corresponds to 0.1 of optical density corresponds to 1 microgram of protein. 0.5 ml of enzyme contains 1 microgram of protein. 1 ml of concentration enzyme contains the following. So you can see the graph concentration of BSA solution against the optical density at 620 nanometer. Now the determination of specific activity of salivary amylase. After standard calibration of maltose, then the protein estimation, we are looking at specific activity estimation. Activator, inhibitor, coloring agent DNS, as we saw in maltose calibration. So you can see the buffers, substrate, activator in distilled water, incubate 37 degrees centigrade, and the addition of enzyme after that, and further incubation at 37 degrees centigrade for 10 minutes. Now the inhibitor is added, diluted enzyme is added, followed by the coloring, the reagent, the DNS. So this mixture is kept in a boiling water bath for 10 minutes. Again, the body and as 520 nanometer. The color developed is read out at 520 nanometer to estimate the specific activity of salivary amylase in this particular experiment. So we are saying about enzymes. This particular enzyme, salivary amylase, is hydrolysis. So when you take enzyme kinetics, it is the quantitative study of enzyme. It measures the rate and affinity of enzyme for substrates and inhibitors. So everyone knows about the famous Michelson-Menten equation. So when you take enzyme activity, we take the measurements of enzyme unit, U, that is the amount of enzyme causing transformation of 1 micromole of substrate per minute at 25 degrees centigrade under optimal conditions of measurement. So these activity measurements and these measurements are very important since enzymes are usually present in very small quantities. So a convenient method of enzyme quantification is the measurement of enzyme activity. Activity is related to its concentration, and enzymes are quantified relative to their activity rather than the direct measurement of concentration. The unit used is the previously said the enzyme unit, or the other unit is CAT is the accepted SI unit of enzyme activity. So 1 catal is the amount of enzyme that catalyzes the transformation of 1 mole of substrate per second. So meantime, we are looking at how we are measuring colorimetrically the color developed in the amylase assay, is the DNS assay, the formation of one pink color is measured at 520 nanometer. Now you can see the calculation. Specific activity is equal to concentration of maltose by molecular weight into 1 by incubation time into 1 by milligram of protein. So you can see the enzyme activity is here, in this case, measured as 0.0361 micromole per minute per milligram of protein.